That's a great question. So we know that immunotherapy works in patients with our esophageal and gastric cancer patients, but unfortunately, the duration of response and the percent of patients who respond, we can definitely improve on, right? So patients who respond often eventually do develop resistance. And what we're trying to avoid is this progression and resistance, both acquired resistance and de novo resistance to immunotherapy. So the way to consider doing it is really to bring in cellular therapies, right? So we've been talking about allogenic and also autologous cellular products. And AgenT-797 is invariant natural killer T cell, iNKT cell product that is really designed to induce direct tumor cell cytotoxicity and indirectly stimulate dendritic cell maturation and depletion of immunosuppressive tumor-associated macrophages and myeloid-derived suppressor cells. So all of these cells have been implicated in therapeutic resistance to immunotherapy in our disease. The challenge with bringing cellular products to the clinic is there's usually a delay in start of therapy. You can't really safely combine them with other agents, chemotherapy and immunotherapy. And so any CAR T type of product that takes time, right? And so to be able to bring in an off-the-shelf allogenic product, which is basically harvested from donors that are healthy, and have these invariant iNKT cells is very appealing. And so that's what we're doing with this study. We know that in this patient population, it's likely that we will need more than just the cells. So AgenT-797 has clinical activity in immunotherapy refractory cell tumors. We have some safety and phase 1 efficacy data with monotherapy administration. So now in the clinical trial development, we're targeting patient population that are chemotherapy and nivolumab refractory. So patients who progressed on standard of care FOLFOX nivolumab, and we're treating them with AgenT-797 iNKT cell with combination of CTLA-4 inhibitor that's Fc enhanced, botensilimab, and also a PD-1 blockade. So it's continuation of PD-1 blockade and combination with targeted agents. So really, we're hopeful that the unmet need of therapeutic resistance will be helped with the study. It's too early to say, but that's the design of the studies to target patients who responded and then progressed on first-line therapy. Yeah, so this study is a treasure trove for several reasons, because it has we're doing an important biomarker analysis, both blood and tissue-based biomarkers. Our patients at Memorial Sloan Kettering are highly motivated, and we have already enrolled and treated 4 patients on the study, even though we were just open in under literally 2 months. What we're hoping to achieve, obviously, is response to treatment, potentially cure in some patients if that's attainable, although these patients obviously come in quite sick and have quite a large disease burden, but also understand that if it worked, why it worked, and if it didn't work, why we think it didn't work. What are the differences between the patient and tumor characteristics from responders and non-responders? And once we have that preliminary data, we will have a clear path to the next study, either a registration phase 3 study or a larger expansion to really bring this agent to more patients. What I can tell you so far, it's been very feasible. We're doing it in the outpatient setting. We don't need to admit patients in the clinic. We do have a highly specialized center. I basically see and treat most of these patients, and then we have a handful of other doctors who do it, so who are all accredited in administering cellular therapies.